next packet, and really the next series of packets, we'll get into membrane structure and cell membrane transport. So the first bit of this is really a review. We're going to review the structure of our cell membranes, and we're going to talk about the important function of phospholipids. Now, we talked about phospholipids a little bit earlier. We talked about the fact that they are these amphipathic molecules. Therefore, they're perfect to act as a part of our cell membrane. They've got the polar head that can interact with the aqueous, inside and outside environment of the cell, and then they've got the non-polar tails, which can face each other. So they're the perfect structure, being amphipathic, to create our cell membrane and to help compartmentalize our fluid compartments in the body, okay? So let's spend some time here reviewing the cell membrane, the structure of the cell membrane, and some of the important components and their properties that are found inside the cell membrane. And this is actually called the fluid mosaic model. Actually, let me create a new slide so I can get a little bit more space here. So this is called the fluid mosaic model. Okay, and the reason it's called the fluid mosaic model is because it is not a fixed or rigid structure. It's actually a very flexible structure, the cell membrane. And so it was named after the physiologist who initially described it, and that's why they're calling it a fluid mosaic model. There's a lot of flexibility and fluidity to the cell membrane that facilitates the function of transport in and out of our cells, okay? Let's draw out the main, sort of the star of the show here, the main structure that forms our cell membrane. So these are these amphipathic phospholipids. Okay. And then remember, there are these double membrane of these phospholipids. So we've got the polar heads facing out and the non-polar tails facing each other, okay? Let's draw in a couple other structures here though. We've got large molecules like proteins that are just spanning the membrane of this cell membrane, spanning the both layers of this cell membrane, right? We've also got some other proteins that sort of studded in between one layer of the cell membrane. And then we've got some other proteins that are fixed on the internal surface of the cell membrane here. We've also got some other ring-like structures, cholesterol. We'll talk about the importance of cholesterol in the cell membrane. What we described cholesterol a little bit earlier, one of our fats, one of our biomolecules. And then attached to some of our proteins, we've got some carbohydrate structures. So some of our branching polymers of glucose. Alrighty, so let's go ahead and start describing some of these structures a little bit clearer. So the first one is the plasma membrane, and it's basically formed by the phospholipid bilayer itself. So the plasma membrane and let's describe some of the features of the plasma membrane. First of all, it's made by the phospholipids. The phospholipid bilayer, meaning that there are two layers of these phospholipids against each other. And this is what gives the phospholipid bilayer its compartmentalizing property because of the amphipathic nature of these phospholipids. Okay. Now, what this does for the cell membrane is it adds fluidity and flexibility. Okay, fluidity and flexibility. So again, this is not a rigid or stiff or fixed structure. It's malleable, it's movable. Things can go in and out. Oxygen, carbon dioxide can diffuse. Fat-soluble molecules can go right through that bilayer, such as cholesterol, steroids, and so on. And so having the structure of these phospholipids and this amphipathic molecule creating this bilayer is what adds some of the flexibility and fluidity to the membrane. Another feature of the membrane is that it is impermeable to water. Okay, really important feature here. It's impermeable to water. And as you would imagine, water is polar, so water cannot exist in between here. It cannot exist in between the fatty acid tails where we have that non-polar hydrophobic area. And so this makes the membrane impermeable to water. And that, as you would imagine, is what helps to compartmentalize the fluid compartments in our body. The fact that water cannot just move through this membrane. So that is the plasma membrane. Let's look at the proteins here. So the first set of protein we have are called integral proteins. They can go right through both layers of the cell membrane. So integral proteins. And if you think about the word integral, it also helps us with understanding the importance of these proteins. So first off, they are transmembrane proteins. Okay, they are permanently fixed in the membrane. Okay, we can have some small examples of one-sided integral proteins, but the main example are the transmembrane proteins. So I'm going to put here as well, we can have transmembrane or one-sided, but the real importance of an integral protein is that the function of the cell is important, is vital, is tied to the presence of that protein. And so while we'll talk about some other proteins that can move around, that can dissociate, that can be recycled and reproduced, integral proteins are fixed structures, whether they're going right through the membrane or they're fixed to one side like this guy is here. The main difference is that these proteins cannot move. They're integral to the function of the cell. If they leave the membrane, the cell dies. The cell is lost, the membrane, dissolutes or dissipates, and the cell dies. So these are permanently fixed in the membrane. Other proteins here are peripheral proteins. So let's draw another, let's use another color here. So peripheral proteins. And peripheral proteins are not so fixed. They're usually on the cytosolic side. And that's this guy, by the way, just to avoid confusion. Usually on the cytosolic side. Okay, so let's add some context here. I forgot to label. So this is the extracellular fluid compartment. In other words, outside the cell. And down here is the intracellular fluid compartment. In other words, inside the cell. Okay, and so this makes this peripheral protein on the cytosolic side, right? So this is a peripheral protein here. So it's usually on the cytosolic side, and then it can easily dissociate or move around. Okay. All right. Let's look at the next structure here, which is cholesterol. And so cholesterol, which are these ringed fat substances in the membrane, also plays a really important function in terms of the fluidity of the membrane. So cholesterol increases the fluidity and flexibility And when we say fluidity and flexibility, it's gonna break up the phospholipid repetitions and allow some malleability of the membrane. So the membrane is movable. It's not this rigid, hard, fixed structure. So it adds some fluidity and flexibility, and that will, in other words, decrease the rigidity or the crystallization of the membrane. Okay, and crystallization is what would otherwise happen if we had really close or densely packed phospholipids. It would create a more rigid structure. And so to break up those phospholipid interactions, we've got these nice ring structures that help add some fluidity to the membrane. It's also going to prevent membrane leakage. Okay, this molecule, being a fat or a hydrophobic substance, is going to prevent any water from moving through the membrane or any sort of leakiness to the bilayer because it's a fat and it's studded in the membrane as well. So it's going to prevent membrane leakage, which further makes the membrane impermeable to water. Okay. So membrane impermeable. And I'm kind of running out of space here, guys. I apologize. Makes the membrane further impermeable to water. Okay. Let's look quickly at the remaining two structures. So these are glycoproteins and glycolipids. Okay, glycoproteins and glycolipids. So essentially, when we have carbohydrates stuck to a protein, this is called a glycoprotein. And then if we got, forgot to draw this here, if we got carbohydrates stuck to a phospholipid, so these are these branching carbohydrate chains on a phospholipid, this is called a glycolipid. Okay, so glycolipid, carbohydrates attached to a lipid, glycoprotein, carbohydrates attached to a protein. And both of these are pretty much bringing about a similar function in the cell. So carbohydrates in general help to create cell tagging and cell identification. This is why they're on the extracellular side. So cell tagging or cell identification. Okay. And so they can be bound to either a lipid or a protein, but they're basically ways that the cell can identify itself in different ways. It can tag itself as a liver cell versus a muscle cell versus a kidney cell. It can also tag itself for destruction or apoptosis. It can signal certain molecules to bind where receptors may be located. So it's just going to help the cell sort of identify itself. It's kind of like the name of the cell, where the cell can communicate with other molecules and substances around it via these sort of flagging or tagging or identifier molecules, either glycolipids or glycoproteins, okay? All right. And I just want to point out here before we wrap up at this point that the central area here in between our phospholipids, okay, this is non-aqueous. In other words, no water, right? This is non-permeable. This is why the membrane is non-permeable to water, because water or polar substances cannot exist within this space. And this is why only fat-soluble, smaller molecules, which can diffuse like oxygen and CO2, can actually get right through the phospholipid membrane. Okay. All right. Any questions at this point?
Yeah, can you go back?
Sorry?
Can you explain the crystallization part again?
Sure, yep. So crystallization is basically when we have many molecules that look alike stacked against each other, and they create a rigidity or sort of a stiffness is another way to say that, because of the dense packing of like molecules, okay? We don't want that in the membrane. And so we kind of break up some of those phospholipid interactions and add other molecules like these ringed cholesterol, which are also impermeable, help the membrane be impermeable to water because they're fats, but it just helps to make the membrane a little bit looser, a little bit more flexible, a little bit more malleable than if we have the dense stacking of same molecules against each other, okay? I hope that kind of added a little clarity there in terms of that rigidity, decreasing crystallization. In other words, making the membrane more fluid, more malleable, or more flexible. Breaking up those like interactions that would create density or rigidity to the membrane. Okay. Any other questions so far? Any other questions about this? Okay, so this is kind of just laying the skeleton in terms of just reviewing the structure of the membrane. What we're going to go on to speak about and introduce on Friday is how we now start to see movement across this membrane. This is really important, building the foundation of us understanding how we can do things like depolarize the cell membrane, create action potentials by moving ions in and out of the membrane, in and out of the cells, essentially. And so just kind of reviewing the structure of the membrane will lay the groundwork for then talking about what can go through the membrane, what cannot, and start to speak about transporters, channels, ion channels that are then embedded in the membrane that can facilitate movement of some other bulkier structures like glucose that cannot go right through this membrane, okay?